Permeability coefficients across monolayers of the human colon carcinoma cell line Cockatoo, cultured on permeable supports, are commonly used to predict the absorption of orally administered drugs and other antibiotics. Here we describe our method for the cultivation, characterization and determination of permeability coefficients of xenobiotics, which are, typically, drug-like compounds, in the Cockatoo model. The experimental procedure is comprised of two parts: the cultivation of the Cockatoo cells on filter supports and the drug transport experiment. The Cockatoo cell line is a continuous line of heterogeneous human epithelial colorectal adenocarcinoma cells. Although derived from a colon carcinoma, when cultured under specific conditions the cells become differentiated and polarized such that their phenotype, morphologically and functionally, resembles the enterocytes lining the small intestine. When cultured as a monolayer, Cockatoo cells differentiate to form tight junctions between cells to serve as a model of the paracellular movement of compounds across the monolayer. In addition, Cockatoo cells express transporter proteins, efflux proteins, and phase II conjugation enzymes to model a variety of transcellular pathways as well as the metabolic transformation of test substances. In many respects, the Cockatoo cell monolayer mimics the human intestinal epithelium. One of the functional differences between normal cells and Cockatoo cells is the lack of expression of the cytochrome P450 isozymes and in particular, CYP3A4, which is normally expressed at high levels in the intestine. Two versions of MEM are required: A. Complete MEM for the maintenance of cells in culture flasks. B. Transport media for maintenance of the cells on filter supports. Cultivation of Cockatoo cell monolayers grown on permeable supports. Trypsinize Cockatoo cells from maximally 90% confluent Cockatoo cultures. Spin down the cells and remove the supernatant. Resuspend the cells in transport media. Place the desired number of filters in 12 well cell culture plates and seed by dispensing half a milliliter of the resuspended cell solution on each filter. Fill the basolateral chamber with transport media. Incubate the plate with the filter supports at 37 C with 5% CO2. Media should be changed on days 4, 8, 12, 16 and 18, and cells will be fully polarized by day 21. Measure the tear values. After washing the cell monolayer with 37C tempered DPBS, with Ca2+, Mg2+, transport media was added into the apical and the basal chamber. Allow for equilibrium for 60 minutes in the cell culture incubator. Tear values, ohm times cm2, were calculated after subtracting the resistance of blank filters. Only those monolayers having tear values greater than 250 ohms times cm2 were included in this study. The Cockatoo Permeability Assay procedure. After determining the electrical resistance for each well, as described above, wash the monolayer and remove the buffer from the filter plate and feeder tray. Transfer the filter plate to a 12-well transport analysis plate. To determine the rate of drug transport in the apical to basolateral direction, add the test compounds to the filter well. Drug concentrations typically ranging from 10 m to 200 m may be used. Fill the wells of the 12-well receiver plate with buffer. Join the filter and receiver plates once all drugs and buffer have been added. Begin timing the experiment. Incubate at 37 C, shaking at 60 revolutions per minute on a rotary shaker. Typical incubation times are 1 to 2 hours. At the end of the incubation, remove a fixed volume, typically 50 to 100 L, directly from the apical and basolateral wells and transfer the volume to a clean plate. The analyses were completed by the use of liquid chromatography mass spectrometry, LCMS, and LC tandem mass spectrometry, LCMS-MS. Mass spectrometry not only eliminates the need for radiolabeled compounds but permits the simultaneous measurement of multiple compounds. The measurement of multiple compounds per assay reduces the number of incubations that need to be carried out, thereby increasing the throughput of the experiments. Apparent permeability was calculated from the following equation. That's all. Thank you for listening.